Okay, so it's just a little walkthrough of how to make the manipulatives and how to use them. So pretty much for all of the files that are in there, you just print them, you cut them out. And actually before I cut them out, I laminate them. And that's kind of an optional part, but all of these manipulatives are laminated. So once you laminate it, you're gonna have two papers, and you're gonna cut those papers and just kind of overlap them. And that's gonna create one long DNA strand and one long mRNA strand. And then on back of this, I just put the magnetic tape or magnetic paper, which is pretty cheap. You can just find that on Amazon. Looks like this. You just kind of cut them up and just paste them onto the back of the manipulatives. So usually I have it on the whiteboard. I also have on the other side, I have my tRNA and my ribosome. The ribosome that's included in the packet's a little bit more simple than that, but you could create a more complex one for the higher levels. So I usually just kind of draw as I'm kind of explaining this. I explain that this is the nucleus. In the nucleus, we have the DNA strand used as a template to create the mRNA strand. So I have our RNA polymerase come in. This says DNA polymerase, but the packet has a RNA polymerase that comes in, makes a copy of this, and then we end up with a mRNA copy, which is this one. This mRNA copy then travels out through pores out into the cytoplasm where it meets the ribosome. The ribosome then goes ahead and reads it. So the ribosome is going to go ahead and read and find the appropriate amino acid in tRNA. So it's kind of important to explain to students that the amino acids are floating around. They're not actually attached to the tRNA yet. So then we just ask the students what matches up with AUG. It's going to be the UAC. So we have UAC. It gets the methionine. So then go ahead and connect that. Then we model that it goes ahead and it brings it into this site. I have trouble grabbing this. So these connects, we have the anticodons connected to codons, and this brings on. And then you would go ahead and just kind of shift it over and then explain that we just go ahead and we just keep adding more and more tRNAs, and they connect. We would bring in the next amino acid and so forth. It's just a little difficult to do with one hand right here, but that's basically the point. You would explain that these two would connect and we would end up with a protein chain that looks something like this. Okay, and it might not be that exact order, but something like that. And there's also, so this would be the teacher model, the large teacher model that goes on the whiteboard, but there's also smaller manipulatives that the students can use.